you don't mind? Yes. Good morning, everyone. Welcome to the last seminar this month. Our speaker is Araz Soman from Heritage Research Facility. I will let Araz introduce himself, and then he will talk about a lecture which is on the screen. So Araz, the floor is yours.
Thank you very much, Devante. And I'm so excited to be here. My name is Araz Solman, and the name Araz Solman, Araz, comes from the mountains of Azerbaijan. But despite the name comes from Azerbaijan, I was born and raised in Iraq, in the mountains of Iraq. And that's where I did my PhD, my honors and undergrad degree in plant biology. And then after I finished my honors, I took a job in Icardi and Aleppo, escaping the war zone in Iraq, going to see a starting a job, and I started in 2009, and that went on until 2012. So that was at the International Center for Agriculture and Dryland Areas, ICARDA. And my role was implementing farmer-oriented breeding to help farmers have a livelihood in a desert-like environment, especially growing barley and wheat. And then after Syria became a war zone, escape again, and this time I went to a big desert known as West Australia. And that's where I did my master's degree at UWA in plant physiology, and then a PhD in plant pathology at Curtin University. So the talk will have a lot of data and a lot of stuff from my PhD, but at the end of the talk there's also things that I do for hermitage in the bacteriology work. And yeah, there's a combination of pathology in fungal pathogen and bacteriology and munging. So you will notice I've changed the title a little to Double Trouble, A Tale of Two Diseases. And I'm referring to handling more than one disease at a time for my PhD and post-PhD working at that. And the reason I'm calling it Double Trouble is because someone asked me a few weeks ago, isn't it enough trouble to handle one disease at a time? And I agree, it's enough trouble to do one disease, to do a PhD on one disease, but doing two diseases is double trouble. But the answer is always simple. When we go to the field, we often see more than one disease at a time. It's rare that we see one disease in isolation. So that reductionist approach of looking at a single disease and single host does not mimic what happens in nature. In nature and in agricultural systems, we often see more than one disease at a time. And these complexities of more than one disease often display properties that we can't capture by looking at that disease and that host in isolation. So that's the whole story. If you want to be bored already, you can escape now. That's the whole story in these slides. Looking at more than one disease, and I'm calling it co-infection. So on the menu today is, I'll give you some understanding, some introduction on co-infection and why should we bother about co-infection. And I'm using the term co-infection in a strict sense of more than one disease, or double infection. And then we'll skip the challenges because there were some videos and I couldn't make them work, but the presentation will go through 3 different steps. Step 1 is the methodology that was developed to study co-infection. And then we look at a survey across the wheat belt of West Australia to understand how prevalent co-infection is. And the third section, there is detailed in vitro and in-plant work on co-infection. And after each section, we'll have a chance to stop, break, ask questions, and interact. So it's an interactive presentation, and we'll have 3 chances to ask questions. And at the end, I'll introduce you to some of the work that we do in mung beans to show you that co-infection can occur across the board in many different species, including mung beans. So you can stop me at any time and ask me questions, it's all fine. And yeah, there's everything for everyone, there's something for everyone, there's methodology, there's a field survey with complex data, and there's a lot of microscopy and transformation using GFP and RFP. So let's dive in. So as I said, the simplified approach of looking at one disease at a time is old, and it does not mimic what happens in nature. But we've studied it, we've used it for 50 years or so, and we've learned a lot. We know more about how plants interact with pathogens, we know more about how plants defend themselves against pathogens using that simplified infectionist approach. But when we go to the field, we often see more than one disease, and that complex interaction can't be captured by looking at each disease in isolation. That's the whole story. What do we know about co-infection? Well, we have a lot of unanswered questions when it comes to co-infection. And what we know comes mainly from the theoretical work of social insect and higher order animal, like human. But I've put together some cartoonish way, and this is published in that paper with that citation. So that shows you what happens to pathogen density, or what's expected to happen in pathogen density when we have more than one disease or one pathogen in a system. Assuming we have two pathogens, orange and gray, and the orange pathogen has a different resource acquisition strategy than the gray pathogen. Say the orange one is more efficient at acquiring resources, then over time we'd expect the orange pathogen taking over, but not completely excluding the other pathogen. So we have a typical case of resource competition. If the orange pathogen has the ability to produce toxins, like a lot of fungal pathogens, then what would you expect over time is the orange pathogen significantly peaks at the expenses of the gray pathogen, and we would have a case of typical exclusion. And if the plant is involved, then the scenario can all change. If the plant targets the gray pathogen, then we would expect a population of the orange pathogen peaking. And if the plant targets the other one, it's vice versa. But if the plant has the ability and have efficient mechanism to defend against both pathogens, then the whole scenario can change. What if we have cases where pathogens arriving at different times or invading different tissues? For instance, invading a spike of a leaf plant, of a wheat plant and a leaf, or invading a leaf and roots, then we have niche in space. So both pathogens invading the plant at the same time by invading different tissues, and their population density changes depending on the environment. So as the environment favors one over the other, the population size increases. And if they arrive at different times, then there's no overlap, there's no need for composition, and the both population can increase over time. So that's what we know, or what we would expect if we have more than one pathogen in terms of density. But what happens to the disease? What happens to the plant? If you have more than one disease on a plant, then the outcome could be of that combined outcome of both diseases could be additive. So 1 + 1 = 2, or non-additive. It could be synergistic or antagonistic. There's a chance that two diseases come and invade the same leaf, and that leaf is a limited source for nutrient and space, and both pathogens could enter into competition, leading to suffering of both pathogens in a typical case of tragedy of the commons. And the ultimate outcome would be to the benefit of the plant. There's also scenarios where the first arriving pathogen can have better chance to access resources and space, and we'll have a case of first come, first served. But if the first arriving pathogen stimulates plant defenses, then the plant could be primed to a second arriving pathogen, and the disease would be a lot less. So these are the questions that haven't been answered, or have been answered in the theoretical space, but there was no experimental or data on support this. And throughout my PhD, I aim to answer some of these questions. And to address these questions, I used a three-way system that involved a wheat as a host and two fungal pathogens. The first one is known as tan spot globally. In the West, and here is known as yellow spot. Some people in the US call it red smudge when it infects seeds, and it's caused by Paranophora triticida. But for the sake of this presentation, I'll call this pathogen PETA or PTR. And I also have a fancy name for the other one, but I'll come to that. The second pathogen that was involved in that three-way system is Septoria nodorum blotch. Some people also know it as gloom blotch, and it can affect both leaf and seed. Both of them can affect leaf and seed. And Septoria nodorum blotch or gloom blotch is caused by Stegonospora nodorum. But for the sake of this presentation, I'll call him PAN. So we have wheat, PETA, and PAN in the system. And why PETA and PAN are a good example for co-infection? Well, the answer is simple. Both of these pathogens are known to infect wheat leaves, and they reduce green leaf area and hence photosynthesis and yield. They both reside in stubble and both stubble borne, so we'd expect to see them in the field most of the season. They also have this polycyclic nature of producing disease, so you would expect multiple cycles of disease throughout the season. So we would not expect difference in arrival time. We'd expect them to produce different cycles throughout the season, and we'd see them all the time. And there's evidence that these pathogens co-occur, but we didn't have data to support the evidence. The only piece of data we had comes from the hypothesis that both pathogens secrete toxin. Sorry for the error here, this E missing. So both pathogens secrete the same toxin, known as toxin, and there's a very famous Nature paper that put a hypothesis together that PETA may have acquired toxin from PAN through a long-term commitment of horizontal gene transfer. So that's the only evidence we have that these pathogens co-occur, comes from the exchange of gene with that gene. These pathogens have very similar disease symptoms. So on the right side, we have tan spot or yellow spot with a classic yellow halo and a small tan region in the middle. And on the left, we have Septoria nodorum blotch with the irregular shaped necrosis. So the symptoms can be similar, but when we have them, side by side, that's tan spot and SNV Septoria nodorum blotch, the symptoms are really similar. So if I were to go and do a field survey in the field, trying to figure out which disease we had at the time, by just simply looking at them, will be nearly impossible. And I'll be making so many mistakes, and I'll be wasting my time because all I can tell is what's on there. That's assuming I can tell which disease is which. And even if I'm able to tell which disease is which, figuring out the contribution that disease is causing to the infection, given that they can overlap in symptoms, will be extremely difficult. And the alternative was to do quantitative PCR or QPCR. And we know QPCR can do the job because we write DNA, we don't worry about symptoms. We know QPCR is fast, quantitative, and it can give us an idea of what's inside the leaf and outside the leaf, which is good. But it can be also tricky, and if you grab me at the end of this talk and ask me why tricky, I can tell you stories. It could be expensive and tricky, but that's what we did. We did sequence alignment, and because some of this work aimed to be in field, we aligned sequences from all isolates of PETA and PAN, so we aligned all these sequences from all the isolates that we know, and then we selected regions to target them for primers, and primers were selected to be of similar size so that they can have the same efficiency. And then to allow multiplexing and add extra specificity, we put probes in the middle of each primer sequence. So in the system, in the QPCR system, we run primers and primers of similar size, of similar efficiencies, and we also had probes in the middle to allow multiplexing. So to test the assay, the first thing we did was doing to see whether our primers are able to tell PTR and PAN, given that the sizes are very similar, and we were targeting a PCR product of about 100 to 115. And we did a PCR, an endpoint PCR with 6 repeats for PTR and PAN, or PTR and PAN, and yes, our primers were able to detect PTR with consistency and PAN or PAN with consistency, so we had no issues with the primers. The next step was, because this work is going to go to the field, we knew that other diseases can also occur with PTR and PAN, and we loaded the assay this time with DNA from 5 different pathogens that are known to co-occur with PTR and PAN, and we also included in the assay wheat DNA. And again, PTR primers were able to detect PTR, so we're running the assay in 2 repeats here. PTR primers can detect PTR only, and they don't cross-react to other species or host DNA, and PAN, same story. So that's the specificity of the assay. Now, how dynamic this assay is. What's the lowest concentration you can detect? And this is simple. This is a QPCR assay looking at, this is singleplex QPCR assay looking at the dynamic range. And again, our probes and primers were able to detect PTR at about 0.05 nanogram or picogram, and the same for PAN. And the efficiency of detection of PTR and PAN is similar. So the R-square is about the same, and the slope and intercept for both of them is comparable. So that's all good, and it's all working fine. The next step was to multiplex them and put them in the machine, and this time complicate the assay by including the other species that we thought they may occur, and they may have DNA underneath on them. And again, as we expected, PTR primers detected PTR, no cross-reaction against other DNAs, and same for PAN primers and probes. The other thing we did is to check how reliable the assay is by changing the ratio of PTR and PAN. So going from 1:1 PTR and PAN down to 1 to 10,000 in nanogram units or in picogram units, and the same for the other pathogen. And we did this because we had no idea what's the expected ratio in the field. So we have to make sure that our assay is not biased toward one when it's in high abundance and the other one in high abundance. So 1:1, no problems, and up to 1 to 1,000, there's no issues. But once we increase the ratio of one pathogen to the other to 1 to 10,000, that's when the assay lagged behind and we will have issues. So if we were to sample a leaf that has this much of PTR and that much of PN, or the opposite. So high amount of PAN and low amount of PTR, then the assay will not be able to detect that pathogen in low abundance. The next thing we did was to load the same ratio assay, with wheat DNA to see what's the lowest disease level or lowest pathogen level we can detect when we have abundance of host DNA in the assay. So we changed the ratio of DNA from 1:1, PTR, PAN to wheat, down to 1:1, PTR and PAN to 200 of wheat DNA. And yes, the assay can detect up to 1 to 2,000, but the issue is once we reach that 2,000, once we dilute fungal DNA a lot with plant DNA, the assay becomes a lot less reliable, and the error bars can be the efficiency of the estimate could be compromised by about 25%. So there's limitation to the assay, and that was the best we could do after probably 4 or 5 months of trial and error, but there's still limitation. But we worked out, in the paper, what this translates to. We would miss the pathogens if the level of disease was about 1%, 1 to 2%. So below that, we will not be able to detect any disease. And the last thing we did was we took that assay to the field to see if we are able to detect both pathogen or one of them from a field sample. And we collected a bunch of field samples from a site. And most of the leaf we collected had symptoms typical of PTR, so with a yellow halo around that necrotic region. But the leaf we collected had absolutely no evidence of PAN or Septoria nodora. We ran that on our QPCR assay, and the result was a bit surprising. Yes, we have a lot of PTR DNA in the blue bar and blue dots, and that contributes to what we see on the leaf. So the total DNA is the black dots and the blue and the black line. But we also see PAN and PAN in very low quantity, despite that the symptoms on the leaf were not present. So yeah, based on this, if I had used visual symptoms, then it would be very hard to complete my PhD. So conclusion of this section is the reductionist approach does not mimic what happens in nature. And in nature, we see more than one disease at a time. And because of the technical advances in molecular biology, we can now study these complex systems easily. Yeah, chance for questions. Do we receive questions online?
Yes.
Any question? I noticed that the year of publication of that Frontiers in Plant Science paper changed from 2017 to 2018. So is it 2 papers?
There's 3 different papers.
Oh, okay, all right.
Oh, there's 5 different papers in this talk, and the last chapter is submitted recently.
Okay.
So there's a lot of data on that one. I shouldn't probably put there, but special audience, I'll put that down.
Okay.
Any questions?
Any other question?
Any PhD student thinking to do QPCR or verify? No? Okay. Okay. So now we have the assay working. We know we can go to the field and collect samples and detect both diseases. So we're confident that we can do it with the limitation that we had. Any question in mind?
Yes.
We got one.
Okay.
Does P have a toxic aging?
Does PAN?
No. I guess it's pyrenophore. So it's pyrenophore, I think. On old, I guess.
Okay. Well, does Pyrenophora aveni has toxate?
Yes, that's the question.
As far as I can tell, this toxate is known to occur in PTR.
Parastagonospora, okay.
Oh, Parastagonospora aveni. I'm not sure. The short answer is I'm not sure. But I know toxate occurs in Pyrenophora drosopharyngeus, Parastagonospora nodorum, and recently there's evidence of toxate in another pathogen. I can't remember the name, but I'm not sure. I don't have the answer right now. I'll look it up.
Okay. So answer will come later.
Yeah.
Yes.
Yeah. So yeah, I'll post maybe Q&A after this.
Yes.
Uh-oh.
It's not working.
No, hang on, I've got a channel. Try again.
Good. So now, after this methodology is being tested and we have strong evidence that we can detect Peter and Pan, my friends, we wanted to go to the field, collect samples, and see how common PTR or Peter and Pan co-infection in the field. And we wanted to know if Peter and Pan ratio that we saw in these graphs that we changed, can they vary by cultivar, by the location of where in the wheat belt, and also by the canopy level, because we also know that PTR and Pan can also be wind-blown in crops. So we had these questions in mind. We went to the field, we went to WA field, and the whole idea of this work was, if we do it and if we see PTR and PAN at the same time, then it makes sense to have them targeted as one complex instead of targeting them separately. So that's the rationale of doing the whole work. And we went to WA wheat belt, and I visited all these sites across the wheat belt and brought back, in about 3-4 weeks, brought back 432 samples. So a lot of samples and a lot of work. But every sample I brought, before I collect that sample, of course I targeted only disease samples, only symptomatic samples, so I didn't take any samples with no disease because we knew the issue with the methodology. If we don't have disease, chances of not picking the disease is high. But we collected symptomatic sample for the purpose of this experiment. And we grouped these samples based on the palette information by their genotype, by the canopy level, and the growth stage of the crop. And these samples, most of these samples fitted within 3 different categories. So we call them wheat pea, Emuroc, magenta. These are the varieties, or others. In the paper, we'll see others. These are other different varieties that farmers grow. And wheat pea has sensitivity to peter or yellow spot, and has moderate resistance to pan or Medora. Emuroc has the reverse, and magenta is expected to have moderate resistance to both of them. So that's what we did. We collected samples, grouped them, and score these samples before collecting them. And then at the end, we brought these samples back, extracted DNA, and run them through a QPCR machine. And then we used the dynamic range of the assay to establish the lowest level of detection. And from that lowest level of detection, we got all these disease parameters, which I'll explain to you in a second. So we have the 3 sites that I'm showing. In the paper, there's other sites, but I'm showing 3 sites here. And we have the overall at the end. We have the 3 varieties here, Emuroc, magenta, and wheat pea. And we collected 2 parameters on these. We were able to collect 2 parameters. Disease incidence, so it's a measure of an infection by any of the diseases, or in other words, if we had positive detection of either or both diseases, we call that sample infected, and that gave us an incidence measure. The other measure was prevalence. So if the leaf had disease, the prevalence refers to which disease that leaf had. And overall incidence was really high, 91%, but we would expect that because we only sampled symptomatic leaves. And to be honest, it's disappointing because we're missing out 9% of the leaves that we were supposed to detect. So there's 9% loss. We know the incidence should be 100%, but there's 9% loss in incidence. But despite that, 94% of the samples that we collected detected for both diseases. So 94% in overall samples, not only on these sites, in overall wheat belt, there were 94% with PTR or peter and pan. Only 6% of the sample had peter only DNA, or less than 6%, and only less than 1% had PAN DNA. So the conclusion from this is co-infection by peter and pan is really high, about 94%, and there's a chance, there's strong evidence that PAN only occurs in combination with peter. The next data shows the score that we did in the field. So as we walked in, we did some scoring, and we ranked these leaves based on the scoring. So we had, again, the 3 varieties, Emuroc, magenta, and wheat pea on the x-axis. And on the x-axis, we also have the growth stage, the ZADOP growth stage, for 24, 26, and up to 55 to 62. And we're looking at visual score, 0 to 100. 100 is dead leaf or completely necrotic. 0 is almost no disease. And we're looking at upper leaf and lower leaves. So from this, as we would expect, the lower leaf on the plants, because they have a longer period of exposure to disease, they have more disease, and upper leaves, the greener, the new, fresher canopy has less disease. And also, the disease progressed forward as we move throughout the season. So starting from 24 to 26 zadok, going to almost flowering. So starting from second leaf, third leaf, going to flowering, the disease does progress, but more so on the upper leaves, on the lower leaves. So this is what happened, what we saw in these leaves, in terms of which disease, which pathogen was abundant. Did we see more PTR? Did we see more PAN? We know 94% of the samples had both, but lower leaves were almost exclusively dominated by PTR. And there's some PTR, there's some PAN on the upper leaves. So the hypothesis here was maybe PTR is strictly stubble-borne, but PAN maybe was wind-blown from nearby paddocks. And also, the fungal DNA data showed us a different picture, a slightly different picture. We had a lot more disease when we visually scored magenta, but when we do the DNA assay, or the QPCR assay, we had a lot less disease on magenta. And there's an easy explanation for this, because magenta can have spotting, and physiological spotting, and that's probably influenced our scoring. So this is another reason why we should be relying on DNA, not on scoring visually, because physiological spotting can resemble disease, and we could be tricked. It could trick the eye. This is another complicated graph. So that's a four-way interaction, looking at the relationship between visual disease score and fungal DNA of both PTR and PAN across the three developmental stages of the plant, and also split by upper leaves and lower leaves. So at the beginning of the season, on lower leaves, PTR is significantly corresponding with disease, and PAN is not causing a lot of disease, is not contributing to the disease, to the total disease. But as you progress through the season, both diseases becomes important, and both on upper and lower leaves. So the start, it's PTR here, causing the disease, especially on lower leaves, not much on upper leaves. So upper leaves, PAN is significant, but as you move, PAN is less significant here. On lower leaves, PAN becomes marginally significant, and as you go to flowering, both diseases can contribute to the total infection. So what's the relationship between visual score and time? Again, the best scoring we got was mid-season. So growth stage, where's the growth stages? It doesn't say here, it's missing. So the best score we got was in mid-season. So the first score we got was less reliable in terms of fungal DNA. The third score was a lot less reliable, and the highest correlation we got between visual score and fungal DNA was mid-season. So here's the conclusion of this section: is PTR and PAN are very common in the repo of WA, and we can give a number from the survey I did. It was about 94% of the samples had both PTR and PAN, but their abundance can change throughout the season. We would see more of PTR at the beginning of the season, especially on lower leaves, but as we progress throughout the season, PAN also becomes important, and it can be more prevalent on upper leaves. Some more chance for questions.
What do you think
think the reason is for your QPCR being the best at mid-season and not towards the end of the season?
Okay. There's a simple answer and there's not a very simple answer. The simple answer is at the end of the season, plants NS, and there's a chance that we're scoring that yellow in the senescing as disease, and it's really hard to tell apart what's the necrosis caused by natural senescing or that's caused by disease. So that's end season. Beginning of the season, a lot of farmers spray chemicals, and there might be some influence from photoburn, phototoxic effect, and that can have influence on results.
What are the symptoms are? I thought you said QPCR, or was it just symptoms you were talking about?
Oh, that graph, let me go back to that graph is a relationship, this one was a relationship between
Oh, it's just visual, okay.
yeah, visual score and QPCR. So if I were to repeat this work and have the control over the wheat belt, I would score mid-season, because that's the highest relationship gain. And it's probably because mid-season the disease is at its peak, and there's a lot less environmental influence.
So you wouldn't just rely on QPCR for your answer?
It's a good question. I would agree with you that maybe relying on QPCR here, even at best, we're getting 69, 70% explained variance. So there's 30% unexplained variance, and we don't know whether that's caused by other diseases that were in the samples at the time. We had a lot of mildew in that year I did my sampling. So there's a chance that other diseases were in effect. And that's probably fine, because also farmers spray for mildew, and once you have dead mildew, that's a necrosis. And that necrosis can be counted as disease when you score. It wasn't only me scoring, there were 12 scorers in that sector.
But isn't that another reason why you would go QPCR? Because all you're picking up is your pathogen, you're not picking up other diseases, other DNA or anything else.
That's correct. So we would assume that this is really reliable because we're only detecting DNA. But why the relationship is not 100% explained? Why? Because there's other things that can influence the system.
So when you did your QPCR optimization, you were trying to see if you can code it, but if the DNA co-occurs, if you can co-detect everything back together. You mix the DNA at different ratio.
Yeah.
Here you are extracting DNA from symptomatic tissue and then detecting. Do you think that DNA extraction can have different efficiency for your plant of fungal DNA?
Yep, and that's part of the methodology paper, which I shared with you. We had controls for extractions. We had both methodology control plus Arabidopsis DNA extraction of pre-optimized Arabidopsis DNA extraction protocol. And we were sure that our extraction method did not bias one pathogen over the other, or did not bias pathogen or plant. Cool.
Yeah, Araz, so you have the relationship between fungal DNA in the plant tissue and the visual score of disease. Do you have relationships between each of those to yield loss?
Very good questions. No, it wasn't part of the objective of this work. And we did have a goal. When we submitted this paper, that was one of the questions, in fact. And I had a go on trying to dig these contracts with farmers to see if we can get yield data. It was impossible. We had a lot of farmers having information about paddocks. So we had contracts with farmers to give us paddock history, variety grown, but we were unable to get yield data from farmers.
Is there any supplemental research happening to look at yield loss of fungal disease in the cereals in terms of disease management strategies and the value of resistance in varieties?
Well, there's a lot. There's a lot. I can refer you to some good ones. If you read what Richard Oliver does for yellow spot, it's some good reading there. But most of this relationship between yield loss and disease are inherently problematic. There's issues with doing quantifying disease because to have a disease-free plot means you just have to use fungicides. And fungicides are also known to be stimulant to green leaf area, to have other properties that can make the plant either yield more or yield less, or extend the green leaf area duration, and hence yield more. So it's problematic to have that relationship between yield and disease. And it will probably be similarly problematic if we do it yield loss and which pathogen you have in terms of DNA. Will have the same issue. I didn't do it, but I can refer you to a good paper by Richard Oliver in Creighton University, who done a really good job on this.
Yes, in the meantime, I exchanged a few text messages with Yupei Tan, who is online, and she has a question through chat. It's a long question, so I better ask Janika to please show the whole question to Araz on the screen. Here, Araz, please.
Here?
Yeah, it's a long one.
Yes, it will be long, but then done then, yes. And then we can move on, yes.
How long is it?
Can you read it, please?
Okay.
Yes.
There's potentially 5 questions here.
5 questions.
And I don't know how to answer all of them, but let me. Okay, so she's essentially saying, did we test against other pathogens like Parasite and open that can also be
UP, yes.
No, we did not. We did not. There's a whole heap of information that I didn't tell you exactly, but we can go through it now. The reason we selected these pathogens is based on ranking in the WFE belt. So we went to GRBC publications, and there was a recent review on which diseases cause the most impact in WFE belt. And our inclusion or exclusion criteria of these DNAs were based on what disease caused impact at that time. And these were the five or six diseases that were on the top of the list.
Okay.
Other diseases were at least of less significant, or they haven't occurred in years. And honestly, we didn't even have DNA for these pathogens to include in the list. We could have gotten DNA, but we didn't have evidence that it occurred. So
Yes, I thought they had a question.
Well, you actually answered my question already even before I asked it.
Okay.
oh, you can ask them, it's fine.
Yeah, because I was about to ask, are these the 2 more important and destructible diseases in Western Australia? Because in the high rainfall zone here in New South Wales and Victoria, the more problematic ones are Septoria triglyceride blotch. You've got striped rots, and you also have the yellow leaf spot and the yellow leaf spot and the leaf rust. So when you were talking about complexities, are there any other studies that it shows, probably not specific to Western Australia, that you've encountered that looks into co-infection and just like what you've done?
Good question. So, is there any studies that have looked at complexes or co-infections in different states or in different parts of the world? Is that what you're answering? There's a lot of work on population genetics of what occurs on plants. So there's evidence of what happens in wheat and in North Dakota. We know the pathogens that occur, but we don't know the proportion. All we know is pathogen A occurs, pathogen B. We have a list of pathogens that can occur at the time. We could have relied, there is information on this even in WA. We know what diseases, we have DNA, we have a study that did community analysis before, pathogen community analysis. And they've done a complete profile of all the fungal pathogens that occur on wheat. But the only issue with that is we know these pathogens can also be environmental pathogens. And if you're just relying on specific sequences, on one specific sequence, the chance of picking an envelope or a plastic sheet from a farm and detecting that some of these pathogens is very high. So without having an idea of how much we have, we can't really tell if that disease was in action or was active contributor to disease, to infection.
There's one point there. For example, if a pathogen is native, it can occur in more quantity and it won't cause much trouble because if it has arrived earlier, it can coexist with plants like for a long time. So I would just say that if it is more, it doesn't mean it is more disease. It depends on the status of the pathogen as well, if it is native or non-native. One can just cooperate with the plants. More does not mean more disease, always.
Oh, true. Okay, so the question is, does more DNA equals more disease? No. Is that what you're saying?
Yeah, that's what your graph says, but in environment it does not mean like if it's more DNA, it means more disease, because if pathogen is native, it can cooperate with plants like, and it won't cause much trouble.
Okay, well, we slightly drifted towards diseases that do not cause infection, so that have other types of relationship with the plants, like peaceful or less peaceful or opportunistic and so on. So that's a whole different field. But what most of the data I presented, I can show you. Hey, so most of the data I'm showing you here is a relationship. I'm not purely only relying on DNA. I'm also plotting that DNA against visual score.
This is fine, but it is like in general terms, so it's not.
Yeah,
Yeah.
In general,
Okay.
there's a chance that you pick, let's say you take a leaf and you detect 10 pathogens. That doesn't mean these 10 pathogens were in action, and it doesn't mean these pathogens were causing an infection. And that's why we did visual score and related that to DNA.
Okay. So in your graph, I saw like 4 or 5 different measures of this, and I'm not sure you have done that. It says incidence, severity, abundance, and prevalence. I'm not sure you have done all of them. Because in incidence, you just went there and you just counted the number of disease plants or number of regions on each plant.
No, we didn't do that. We didn't do that. So incidence is a leaf that has an infection, either PTR or PN, using QPCR. So that data that I showed is purely in the table at the very beginning. Just got another question. Okay, I'll come back to that question in a minute. So this is purely DNA, relying on DNA. So when we say 91% incidence, means 91% of the samples that are collected either had PTR, PAN, or their combination.
Okay, so what's prevalence then?
Prevalence is of this 91%, which one was PTR, which one was PN, which one was co-infected. And these numbers are the proportion, and the number in parentheses are the number of samples that were in that detection. So there's 8 samples, 66% of them were co-infected. Did I answer your question, or?
In abundance and
I didn't. Abundance is.
I saw the graph in abundance and severity as well. So what's that? Because it's very hard to do everything at one time. I'm just saying that it's probably impossible. If I saw in the graphs like abundance, severity, and prevalence, incidence, it's a big words. Like it's a very hard job if you do it at one time. So I don't see the methods, but I see the graphs. Like when I say prevalence, then I say incidence, then I say severity, and then I say abundance.
That's abundance. This is abundance. So this is in nanograms per 40 milligram leaf tissue. How much PTR, how much PAN we had. There's missing something, or how much disease, how much fungal DNA. Sorry, how much fungal DNA we had on these leaves. And this is abundance of each disease, abundance relationship. So score and nanogram of DNA of PTR and PAN. So this is abundance of disease, how much fungal DNA, total fungal DNA, and this is abundance of PTR and PAN and its relationship with visual score.
So we also had one more question from Threeway, because I only saw the first part of the question. The QPCR is targeting the TOX8 gene, I guess. This is what she wrote.
The QPCR is designed to target TOX8 gene. No, that QPCR was not tied in the toxay gene.
Okay.
And the simple reason, if we had targeted toxay gene, we would take toxay of both pathogens. Toxay in PN and toxay in PPR is nearly identical.
So which was the target sequence that you used? ITS?
No, it's not ITS, it's a specific, the PPR gene is downstream of toxay, and it's about 300 on the base region. Details in the paper, I think
Yes.
show you the paper. And PAN gene is a unique gene that occurs in all PANs, PAN isolates, and it's not known to occur in other species.
Okay. So these were species-specific sequences.
Yeah.
Is that 100 and something?
There was another question.
Yes.
Is this your paper? Said it was toxide.
Yes. So
No,
maybe
it's downstream of toxay.
downstream of the toxay gene.
Yeah,
Yes,
downstream.
I think we can continue these detailed discussions later on. Maybe you can move on to the third part of the talk. And it's so good to see that it's really interactive.
I like it, the interactive.
And I'm sure people will stay here and we will have a very good discussion. Yes.
What's that again?
No, sorry.
So now we did the survey and we know PTR and PAN, the Coinfection is common. The next step was to do a detailed study to bring these back to the lab, to the glass house, to the growth environment, and see what can we get from this Coinfection. So objective of the next few slides, and there's so many slides, is how do PTR and PAN, or how do Peter and PAN interact to cause disease in the plant? So looking at the fate of the infection, we have both of them. Does the inoculation sequence affect the outcome of the infection? And what are the mechanisms, if there's any, that regulate the interaction? And what regulates that niche that we see early season and late season, early season being Peter dominant, late season PAN dominant. So these are the objectives of the coming slides, and we started from the very basic by just doing plate assay, nothing else, having PTR and PAN and plate assay. So we have PTR plus PAN, so this is resembling co-infection, but it's not strictly co-infection, with no distance, so we put them right side by side, and then we have plates with so this is 2 repeats, I'm showing 2 repeats, plates of PTR only or Peter only, and plates with PAN only. And as you can see, 3 days after putting these pathogens next to each other, the plate that had both diseases grow much bigger than the plates that had one disease. Okay, so I have even numbers, so 4 times when we have them combined, the growth is 4 times or 3 times, about 3 times more of the single pathogen only.
So is that both diseases though? I mean, I can't tell the difference between the two.
Good question, really good question. I did QPCR assay, and no, the answer is PTR. So Staphylococcus aureus remain in the middle, stuck, and what grew is Peter. 6 days after, Peter taking all the space when we had next to him PAN, but PAN on its own is a lot smaller, and PTR on its own slightly smaller. Now, what if we put them separate on 2 different ends, distal ends of the plate? So we had PAN, same isolate, on the 2 ends, and Peter on both ends, and then we had the switch, Peter and PAN, Peter and PAN. When we had Peter and PAN, even on distal ends, Peter grew a lot faster than when we had them when we had Peter next to Peter. So interspecific interaction is happening there, and that's 6 days after. Peter took over most of the plate, PAN is restricted.
But there is an inhibition zone between PAN and Peter. Probably the red arrows indicate the inhibition zone.
Yeah, I call it aggression zone
Yeah.
or confrontation zone, but we will come to this. There's detailed GFP, I think detailed GFP, RFP with real-time microscopy showing what happens in that aggression zone.
It's not just simply overgrowing one another. It's not just simply overgrowing. It's something is happening.
There's a lot happening, and if there wasn't, I wouldn't have a PhD right now. The next attempt was to transfer PETA and PAN by tagging them with different fluorescent proteins. So tagging PAN with GFP and tagging PTR with RFP. That took a lot of time, but the result of this at the end was I was able to transfer PAN, but I was unable to transfer PTR. This is product transformation, and it was extremely difficult. I did do transformation at the very end, and I'll just quickly show you a slide. So I did do transformation, successful transformation of both pathogens, and that's a slide showing PTR in red and PAN in green, but that data went to the submitted paper, so I can't show most of that data. But yeah, nonetheless, for the sake of this presentation, I transferred PAN, and PTR is tagged with immunofluorescent protein. And if you want to ask me how I did that, we can have a chat at the end. So we grew PTR and PAN, or PTR and PAN on a slide, on a basic, simple slide. So we caught the slides with agar, and we had them installed or grown on either end of the agar slide. And we let them grow until they start overlapping in the middle. And we start calling zones. So that's that end, the far end is PTR zone, the lower end is PAN zone. And in the middle we call it confrontation zone. And when we do microscopy, just optical microscopy, that's what we see. We see PTR at that end, we see PAN at this end. There's no pathogen growing beyond its zone, at least in the far sides. And when we tag PAN with GFP, that's what we expect. And when we do that immunofluorescent staining on PTR, that's what we see. Okay? In the middle, there's a lot of things happening. There's curling, we would expect them to have a separation zone, but in fact, what we saw microscopically was these pathogens have the ability to join and intermingle and cause these, even join their hyphae. And I did have a video, but I had to remove it for some reason. I can show you in a video after this if you want. I can show you these pathogens have the ability to join and fuse. They can fuse the ends and do some exchange stuff. But I can show you at the end of this presentation if you like. At the point of contact, these pathogen the green is PAN, the blue is PETA, and they can also bridge the hyphae and interact and come close to each other with no antagonism, with no aggression, and they can form these network. And this is where when I transfer them with RFP and GFP. So PETA and PAN, by the way, this is a real-time microscopy. So videoing with a confocal microscope, with a big multi-million-dollar confocal microscope. And we're looking at PTR in red and PAN, or PETA in red and PAN in green, GFP and RFP. And these two pathogens can grow. PTR will take over, but they can grow peacefully, no aggression, absolutely no aggression in plates. And in fact, what you see here, that yellow is just an overlap of red and green. So when they come in contact, I have a video of this, when they come in contact in plate, they have the ability to regulate the contact by spatial separation. So one of them goes deep down in the agar, and the other one is staying at the surface. And we did a video of this, a 3D video of this. The next step was to do detached leaf assay, so to expand on the complexity a bit by doing detached leaf assay. And what we did this time, we had 4 genotypes, and we infect them with Peter, Pan, their combination, or no infection at all. Okay? And what we're looking at here is how quick the symptoms appeared on the plant when we had these different regimes of infection with these different varieties. So all the time, almost all the time, when we had Peter and Pan together, we had quick symptoms. The symptoms appeared 3 days post-infection or post-inoculation. And when we process these leaves on a QPCR machine, all the time, in all the varieties, we detect Peter, no evidence of Pan. So we do get quick symptoms when we have them both together.
I'm messaging with people on Zoom, sorry.
No problem. So we have them both together, we get quick symptoms, but that symptoms are only corresponding to detection of PTR. PAN is below detection level. This is when we play with the arrival sequence of the co-infection. So same variety except magenta, because magenta was not infected. We were not unable to get infection from magenta. And we had number 1, Peter and Pan at the same time. And 2 to 4 is changing the sequence of PAN to either 24 hours after, 48 or 72, and vice versa. Okay? And again, it looks like we get a lot more disease when we start the infection with PAN. So when the infection starts with PAN, PTR takes over and it causes a lot more disease. This is the data from what I showed you earlier, but over time. Okay. So whenever we had PTR and PAN at the same time, that's the green bar, we had quick symptoms. And here's the figure. So we had PAN, PT. Oops, there's a mismatch here. That's the orange one is PTR, it's not PAN. So blue is PAN, orange is PTR, and the green is PTR plus PAN. And when we have them both combined, we get a lot quicker, a lot more disease and a lot quicker disease. And again, that's what we saw in the QPCR machine. So we get, when we have them combined, PTR and PAN, we get quick disease. But all the time, when we had those 2 pathogens, what we detect is PTR. So most of the disease that we score or detect in the machine is corresponding to PTR, and no evidence of PAN. But when we change the sequence of the infection, so when we have PTR and PAN at the same time, we get quick symptoms and a lot more disease. And when we have PAN and then PTR 24 hours after, again, we get quick disease. So when we start the infection with PAN, we get a lot more disease. The reverse order, if we start the infection with PTR and then PAN, we do not get the same results. So there's some magic of which pathogen start the infection, but we don't know why. Well, we won't know that why in these slides, but we know some answers and some anything. Again, here we're looking at abundance. So fungal DNA abundance in these co-infected leaves versus the singly inoculated leaves. The green lines are co-infected, with the empty symbols is PTR, and the other one is PAN, and the blue bars are single inoculation. Okay? And consistently, except in magenta, consistently PTR takes over time, while PAN stays nearly at 0. So from this graph, if we have PTR and PAN on the same leaf, PTR takes over time, and that diminishes PAN co-infection. This is what happens if we change the sequence. So if we had both pathogens at the same time, almost all the time, PTR takes over. Even when we delay PTR by up to 72 hours. So if we do PTR now and PTR 3 days after, PTR takes over. But we only see PAN taking over the infection if we delay PTR by 72 hours, by 48 to 72 hours. So there's a PTR seems that it's more aggressive and it's taking over whether it's starting in the beginning or late. But PAN can have the first come, first serve. If it's
Do they infect on the same growth stage?
yes. So PAN becomes a bit more dominant if we delay the infection of PTR. So the next was to see the interaction in planta. What happens in planta if we have these two pathogens at the same time? And the first thing we did was to do an inoculation with PTR and then inoculation with, so there's no co-infection, we're just doing inoculation with PTR at a time and then PAN at the other time. And we see some difference in the way PTR and PAN infect the plant. Almost exclusively, Peter entered the plant through stomata. And that's, we're looking at confocal microscopy on the left, and both other pictures are scanning electron microscopy. So almost all the time, I think we did 100 infection assay, and 99% of the time, PTR enters the plant through the stomata. And that's, again, that's a stomata picture, and that's PTR with infection that entering the stomata. But PAN, 98% of the time, has a different strategy of entering the plant. And that's, we're talking single infection, not talking both. When we do an infection with PAN, PANs enter directly through the leaf, so it generates that infection peg, and it directly, through a pressure mechanism, directly poke through the leaf. So it does not enter the stomata, it chooses to enter directly through the leaf cuticle. What if we have them both at the same time? So when we do co-infection, this is what we see. And this is again confocal microscopy with PTR tagged with that magenta or that blue protein, and PAN will be green, but I'll show you PAN in a minute. So when we did co-infection, we did not see evidence on the leaf surface, we only see PTR on the surface. So PTR enters through the stomata and immediately very quickly, rapidly occupies the leaf surface. Whereas PAN? Well, PAN is really deep down in the vascular bundle. So we repeated this, and we have a video of this, and probably show you the video after this. The way these two pathogens regulate their niche is by occupying different niche within the leaf. So they can co-occur, but they're not strictly at the same place. PTR is on the surface, PAN is deep down, and this way they can both coexist, but they don't occupy the same niche. If you're not convinced, what we did then after this, we said, now, what if we restrict them to a specific spot? So we infect them at the same time and in a single spot. So we put a ring around the leaf, we put a small ring here in the middle of the leaf, on detached leaves, and then we put PTR and PAN right in the middle. So we did spot inoculation with a pipette, equal concentrations, and in the middle. And then we took tens of leaves and dissect these leaves after 7 or 9 days on a 1 to 3 centimeters from the center. So we took a sample 1 centimeter, 2, and 3 from both ends to see which pathogen were able to extend beyond the inoculation spot. And this is what we see. So PTR or Peter has the ability to grow and go all the way to the edges, but PAN gets stuck in the middle in a dense mycelia of PTR and does not grow beyond the inoculation spot. So the conclusion of this was, yes, they can coexist, but PTR has the ability to trap PAN in a dense mycelia, and hence PAN cannot grow further.
Is there any reason for that, like pen cannot grow? Because it's very interesting.
Why they can't?
Yeah.
No, but from this, I can tell you, PTR has the ability to grow dense mycelia.
Yeah, and pen can, but is there any reason? It was very interesting.
Not sure. I don't have an answer. I don't know why. It might be to do with speed of growth. It might be to do with aggressiveness and a whole bunch of reasons that I'm not sure about. But this is what we know. We know that they can co-occur, but PAN is trapped in the dense mycelia if we force them to stay together. And this work is all repeated. These parts are in my PhD, but they're all repeated in a high resolution images and high resolution microscopy. Now, the last thing on this. What if we bring a friend in the equation? What if we bring cousins and close relatives? So what if we bring another PTR in the system? So another isolates of PTR that are also known to occur in the field. So what we did in this assay, we're measuring the recovery of fungal DNA, so how much of that pathogen DNA we can recover, and this assay relies on toxA. Here's when we're relying on toxA, but toxA gene. So what we did here, we had two isolates of PTR, one produces toxate and one lacks toxate, and another isolate of Staphylosporidorum. So SN15 is PAN, and these are PTR. When we have two friends, the two friends drive the enemy out of the system over time. So PTR and PTR can gang up against PAN, and PAN over 12 days, it can diminish in the system. And this is broth, only on broth. We did not do that on plantain. And if we change, so we have SN15 and SN7. So these are two PAN isolates, and we put that with PTR isolate. And yes, PTR can resist a bit at the beginning, but then both PAN isolates have the ability to drive PTR out of the system. So there's two brothers against a cousin, two cousins against their neighbor, I have no idea, but this essay was criticized so heavily, and I did not end up putting this in any of my pieces because I'm relying on toxA, and there's a heap of other issues that you can criticize. But we didn't have any other way to do it. The only way you can do it is relying on SNPs differences in these patches.
So this isn't the broth. Even if the other one dies, wouldn't you expect to get some DNA out of it? Or?
Yep, you'd expect.
But you have zero.
Because this is broth, so we took out of the broth and incubated on plates. Well, grow on plates, why not QPCR? Otherwise, if we only took the broth, we will measure the concentration of toxin because it's culture infiltrated. So this is the conclusion of this chapter, or this section. PTR have the ability to outgrow PAN both in vitro and in planta, but there's no antagonism. The only antagonism we see, and we have limited evidence for, is when we have isolates, or we have kinship. When we have PTR and PAN at the same time, we expect more disease and rapid disease, and it seems to be that rapid disease and more disease almost all the time is corresponding to more PTR, and PTR can benefit from being with PAN. There's evidence that PTR and PAN invade different tissue within the leaf, so they specialize in space in their niche, and hence there's no overlap. But if we force them to stay together in the same spot, the evidence is PTR can trap PAN, and PAN does not grow beyond the infection spot. a burning question. So there's a lot of questions there.
It was very interesting study, thank you. So PTR grows better than basically inhibits the PAN. But do you have any reason? Do you think if you used other isolates of PAN, you would get different results? Or how did you choose your isolates?
These isolates were selected from the field, and they were selected from 2 different fields. I did not include isolates in this work. Short answer, I didn't test. There's a chance there will be different outcomes if we include different isolates. But the reason we included these isolates was we wanted isolates that shows no history of interaction, because we also know that these pathogens, if they stay together for long, they can form bridges, they can exchange genes, and there's a whole paper on horizontal gene transfer. So if we take isolates from the same field, these isolates, there's a chance they will have already formed some bonds, some form of bonds. So we selected isolates. PTR isolate comes from humerus, and PAN isolates come from gelatin, 2 sides of the wheat belt, and we included them in the analysis. I did not test effective isolates. The exception is the broth to plate assay, that the last graph I showed you, and we didn't do it for paper either. But it's a good question.
I think it's a big discovery that actually PTR benefits from PAN infection. And I think that's really, really exciting. What if we just talk about all these things during the tea after the seminar?
Is there an online question?
Do I have?
In the meantime,
Do I have 5 minutes? 5 minutes.
yeah, just a second. In the meantime, a few people who are online, they wanted to talk to you, maybe by Zoom after the meeting. So there are so many questions. So I suggested them to wrap up your presentation with the monkey disease part.
Yeah.
And then maybe we will establish a Zoom link, and they can ask their questions directly from you, just like people from the audience, if this is all right with you.
Absolutely, absolutely. We
Yes.
can do a Q&A session, or we can do
Yes,
an online interactive session, or you can grab me, give me a call, and ask me any question.
yes, yes.
So, very quickly, in the monkey world, this is what I'm doing in the monkey world. We're looking at 3 different diseases in monkey, and these are bacterial diseases. The first one is halo blight. So it's called halo blight because it causes these necrotic region with a yellow halo. That's what's called halo blight. So that's a bacterial pathogen, seed-borne, prevalent in cool environment. It's highly destructive pathogen. It can cause yield loss up to 30, 40%, even 70% yield loss. So that's the first pathogen we are in our hit list on that. That's work in collaboration with Cole and other colleagues. The other disease we're looking at in DAF is tan spot, and this is a disease more prevalent in the dry, hot environment, so end of the season maybe, and it causes these yellow patches with irregular shape, chlorosis or necrosis. And again, it's a bacterial pathogen, seed-borne, and what is it? And it's prevalent probably in end of the season. The third disease we in our hit list in DAF is powdery mildew, and everyone is familiar with powdery mildew with the classic powdery-like symptoms. And powdery mildew occurs mostly in the middle of the season, and it's a disease that relies on suitable human environment, cold human environment. So these are the 3 diseases that we now are watch list in DAP, and we have a lot of work, and we did some work on them, but I wanted to show you this. And the reason I wanted to show you this was co-infection occurs in wheat, but there's evidence that co-infection also occurs in monkey, and I'll show you what I mean. Here's a leaf that I collected from Redlands, Brisbane, and these spots highlighted by the red arrow are halo blight. These big lesions highlighted by the black arrow is tan spot, and that's mildew. you go. So that's if you still dark co-infection, that's a good example that 3 diseases occurring on the same exact leaf. And we did some work on testing some genotypes this year. Last season. And what we see the relationship between halo blight and powdery mildew is a typical inverse relationship, and we have a typical easy answer to this. Halo blight is a more necrotrophic lifestyle, so it kills host tissues to get nutrients, while powdery mildew relies on green tissue, and if you have more dead tissue, you have less powdery mildew. So there's an inverse relationship in halo blight and powdery mildew. But we have, with some brilliant minds called Douglas, sitting right here, we bred a promising variety that has good resistance to powdery mildew and halo blight. And hopefully, we have more in the pipeline called for other diseases. So that's last thing. These are the people that I acknowledge for the work I did at CCDM in Kelvin. So Richard Oliver, Mark, Fran, Caroline. And that's the very special person, our collaborator, John Hamlin from EWA, from Perth. And he was also a manager at Ricardo. And he is the beautiful people that I work with now at DAF, called Rennie, William, and Katie. Thank you.
Thank you very much. I'm just wondering if we should stop the recording at this stage. I am asking this question from Evan, who takes care of open pathology. What do you think? Probably we should stop recording here.
That's fine.
Yes.
We can edit.
Yeah, and then we will continue our discussion, obviously in the room and